Good afternoon, everyone, and welcome to Rodale Institute webinars. It is my second show this year, and today we have a very exciting project we are going to talk about. Joining me from Strauss Research Center, Dr. Junjun Khan and Dr. Diana Oviedo Vargas. And together we are going to talk to you about soil microbes and bean nutrients in the vegetable systems trial, comparing side-by-side organic and conventional systems. I may shut off my camera during the presentation to be more focused on the content. So black beans and green beans are considered many of those plants that are accessible to many people, easy to grow, especially in tilled systems, and they are known to be rich in minerals and proteins. However, when you look in the literature, you will find several gaps, especially when we are comparing the nutrients or the microbes related to the black bean or green bean in organic or conventionally grown. Variation in the diversity and richness of microbiome is not known. And also, what would these microbes do with the enzyme activities, how they change with time, especially under different systems or different varieties. These are gaps in literature. We would like to fill those gaps with our knowledge. But most importantly is that would those soil microbiome affect the quality of the nutrient, which is a big question to answer, but we are not going to answer it directly today because we still have one more year of work to add to the knowledge that we have been gathering. So moving on, this project started in the vegetable systems trial in 2021. And those who do not know about vegetable systems trial, we have put several webinars before specifying the details of vegetable system trial. I would like you to go back to those to learn more about it. So we planted green and black beans in 21, 22, and 23. And we put that in reduced tillage in two systems, organic and conventional, and in those with 4 replications. Here you could see that on the right side is the organic system where we reduce till we roll crimp the cover crop, but in the conventional we apply herbicides and then we seed with the monosem planter. The project also, we collected soil samples and we collected those from the 0 to 20 centimeter 3 times a year in 2022 and 2023. And we did that at 3 times in the season. And mainly we wanted to assess the community, the microbial community, the diversity, richness, and we wanted to know if these microbes have different enzyme activity. So we collected samples in the initial, which is at the cover crop season in the spring, then mid-season, which is at the flowering stage of the crop, and then at harvest. Along the way, in the nutrients, we have collected the data from 2021, 22, 23 is still in progress, and we haven't analyzed them yet. But today we will talk about the vitamins B6 and C. We'll talk about minerals, mainly the macros we are looking into. But we assessed also micronutrients, proteins, which is the crude protein, and ergothioneine, which was only assessed in black beans. And because the lab that is assessing ergothioneine is still developing the protocol for the green beans. So hopefully by next year, we have data for green beans and compare both of them together. Also, we collected samples and we assessed for amino acids and anthocyanine in black beans. These are still in progress. And so anthocyanine, we are collaborating with Purdue University, who are working with us, Dr. Lavanya Radvari, on defining the anthocyanins. But amino acid and anthocyanins will not be discussed this time due to the limit of the time, but we have plenty of data to show you. So let's look at the vitamins as a result. So here what we have on the y-axis is the amount of vitamin B6. And on the x-axis, we have green beans and black beans, which is the bean variety. In the blue is the organic bar, and the red is the conventional bar. You could see that there is no difference between organic and conventional regarding vitamin B6 concentration, but there was a difference in the black bean, and usually looks like in this over 2 years, the trend is the same, that the black bean has more organic, in the organic system, vitamin B6 about than the conventional. And you could see that the beans, the green beans, have about 30% more than the black beans in vitamin B6. Going into vitamin C, you could see here that the black bean has really negligible, extremely low amounts of vitamin C, but we assessed vitamin C because we knew green beans has vitamin C. And again, here you could see that the organic is in orange, is statistically different from the conventional in the vitamin B, in the vitamin C. Moving on to proteins, we find here that both the green beans and the black beans, both of them in the organic system, are greater than the conventional. So both of them are affected by the system, by the cropping system. And you can find that the black bean has more than 45% of protein than the green bean. This trend was followed the same with nitrogen. So the protein was higher in the black bean as well. Here we see the nitrogen is higher in the black bean than the green bean. And also we found the organic system is greater in the amounts of nitrogen in it, about 50% than green beans. Moving to the phosphorus, similar scenario, we found more in the black bean than the green bean, but the green bean was more affected between systems than the black bean. And if we look at the sulfur, is also the sulfur in the black bean is more than the green beans, which is about 2 times greater, and it is providing a lot of sulfur in this system. Again, organic is more than conventional. When we look at potassium, though, we see the green beans showing in the organic more potassium, but there was no significant difference in black bean. But again, you could see more of the green bean showing more potassium content than the black bean. Ergothioneine, we talked about ergothioneine at the beginning of this presentation, which, let me define this. It is a potent antioxidant. It is produced in the soil, but not by the plants. And those ergothioneine, it's like a amino acid. It is a rare amino acid. It was firstly found in areas where mushrooms are growing. And so we are looking into ergothioneine as an amino acid to see if it is available and at what concentration, especially when we are looking into organic versus conventional. What it helps ergothioneine, and it is for human purposes, it is helping the cognitive functions, reduces inflammation. It is known that it reduces Alzheimer's disease. And when we compare here, this is black bean ergothioneine. When we compare the amounts of ergothioneine found in black bean compared to kidney bean, we found that the organic system, the black bean, has nearly similar amounts as kidney beans. Now, not all crops, all grains or vegetables have enough ergothioneine, but mushrooms have higher rates of ergothioneine. So as I said previously, we cannot show you the green beans if they have any ergo because it has not been done before. But Dr. Pitzer at PSU lab, he is developing the protocol, and we will share this information by next year and compare those information. In summary, for my part here, the black beans are richer in proteins, nitrogen, phosphorus, sulfur, and ergothioneine compared to the green beans. Green beans, they are richer though in B6 and C and the nutrient potassium. We found also that the nutrients are mainly greater in the organic system than in the conventional, and including both beans in your diet can provide immense amounts of vitamins, minerals, and proteins. And so now we will share this. I will put this presentation down, and Dr. Khan will start his presentation starting from this slide.
Yeah, well, thank you, Gladys, for the introduction. And my responsibility for this project is working on the microbes in the soil samples Gladys collected from the vegetable system trial. And probably everybody knows the microbes, they are critical for the soil health, right? And don't need to mention in the soil, even you have 1 gram of soil, you have about like 1 billion microbes in there, including bacteria, archaea, and fungi, right? And they are critical in the soil health. And I listed 4 reasons here, and they can improve the soil structure, and they can form the microaggregates with organic matter. So that's how you have your soil structure. And when you go outside and take a look at your soil, you say, oh, this soil is very different from there. You know, part of the reason is the microbes are different. And also in the soil food web and all the microbes, they are the foundation, which means they serve as the foundation of the food web. If you have the worms in there, you know, they rely on the microbes. And more importantly, the microbes, they are degrading the organic matter for the plants, right? Before the plants can take up all the nutrients, and the microbes are really working hard for them. And then lastly, just with the microbes in the soil, they enhance all the nutrients, including the carbon, nitrogen, phosphorus, and the sulfur, et cetera, and to help the plants, the uptake, and also, of course, speed up the cycling. Okay, so I probably gave a little bit background about what we did, because in this day still, we have really advanced technology and try to understand the microbial structure and the function, but still there's more than 99% of the microbes from the natural environments, they are not readily cultivated in the lab. In other words, we cannot bring them in the lab culture. There's no way for us to understand or study their function. So that's why we rely heavily on the molecular tools in these days. And I have a flow chart here. So we have the VST after we collect the cores, and we extracted the genomic DNA. And then after we quantify them, and then we can conduct a whole series of molecular analysis on this, which means we amplify the genes we targeted at, and then after run this high throughput sequencing, so we're able to identify what kind of microbes in there. As I said, there's only less than 1% of the microbes can be cultivated in the lab. That's why we rely on these molecular tools, which can tell us at least we have this gene called the ribosome RN gene, which every single cell contains that gene. So we're able to ID what kind of microbes in there. And to me, I think the first question, if we were trying to understand the microbial role in this soil health, and we need to understand what kind of microbes in there, right? And was thinking about with all this huge amount of microbes in the soil, I guess that's the first question we try to understand. So after that, and we know the microbes in there in the soil, they're so diverse. I mean, I show a curve here, and this is the y-axis show you the relative abundance of a certain species, and the x-axis can go all the way. This is how many, they call it OTU, but you can think about this species, right? So when you have like a really high diversity samples, which in this case, you see some of them are pretty abundant, like here rank over here, only a few species are pretty high abundance, but most of them are in this tail. So really, there's millions of species are in there, all the way they're sitting here. So which means they are not as abundant as the others. But on the flip side, if you have a low diversity, you can see the tail is very, very short here, right? And fortunately or unfortunately, we are facing the soil, which is a very diverse environment. In other words, we have tons of microbes in there. And that's why for this project specifically, why we try to identify what kind of a bacteria, archaea, and also fungi in these different systems of the vegetable system trial. And we identify with this analysis, we were able to get a whole lot of sequences out of it. I mean, I give you a number here, about 1.276 million sequences per sample. That's for bacteria and archaea. And also for the fungi, we have 1.185 million. So we really need that coverage to understand the diversity. As I said, the soil environment is extremely diverse. And we try to cover the small tails like here, right? You're thinking about, if you're very high diversity, you want to know what those microbes do. And I don't want to get you wrong, those microbes, they are not necessary to be high abundance, but they might be critical for the soil health. So that's what we did. And we only have, as Gladys mentioned, we only have the 2022 data here because we just collected 2023 and we get those ready and they are to be sequenced soon. So we were able to analyze them together and put them with the data from 2022. So let me show you a little bit data here, the results from 2022. Again, as I said, on the left side is the bacteria archaea diversity, and on the right side, I show the richness. So the difference is the richness you can consider as the how many species are in your each sample. And the left side is the diversity. You may ask me, like, what's the difference? Yes, of course. So the richness is just a number, right? You can see on the y-axis here, you can consider those as the species number. But the diversity we use is a Shannon index. And the difference is the diversity index, we include the evenness. In other words, like how relative abundant of each taxa. It's not only like you only have 100 species, for example, but if they are evenly distributed in the system, that means that they're more stable. In other words, if you have a higher diversity samples or the environment, you're more productive, you're more tolerant, and you are more resistant. So in other words, on the flip side, I mean, it's an extreme example. It's like you have a 100 species, but only a couple of species are super dominant, and all the others are like a very, very rare, then that system is probably not stable. And that's why we have this diversity and the richness listed here. And you can see from the beginning, again, T0, that's the pre-season before we plant the beans. And then the flowering season, you see both the diversity for the green bean and black bean, they are lower. But the blue bar here shows the organic system, you see it's still higher. And we have, Gladys mentioned this, and obviously we apply the herbicide at the beginning of this season or before the flowering season. So that might explain, actually, talking about that, I think Gladys and I have a project working on the pesticides, how the pesticide impact on the microbial community structure, but that's out of scope of this project. Anyway, we have the pattern, like the trend is like a beginning, you have pretty high diversity, and then the flowering season, you have lower. That might be part of the herbicide, as I said, and it might be the seasonal. That's why we want to have this flowering season and also the harvesting season. When we get to the harvesting season, you see the bacteria diversity are getting back a little bit, but still most of the time you see organic, they contain higher diversity than the conventional, which is the yellow bar here, or the brown bar. And the richness, it also shows kind of like a similar kind of pattern, but it's not as obvious as the diversity index. So you see from the beginning T0 here, and then after you apply the herbicide, we get a little bit lower the flowering season and coming back a little bit. So again, this is the richness, difference, and diversity. And we, most of the time, we try to look at the diversity rather than the richness. The next slide, we try to show the fungi. I mean, we are, in addition to the bacteria, archaea, and we look at the fungi. I mean, probably everybody knows the fungi is very important, right? From that relationship with the plants. They didn't show up the similar pattern as the bacteria. So you see from the beginning all the way to the harvest season, they maintain about the same level of the diversity. Of course, you may notice that their diversity is not as high as the bacteria, right? And again, the richness, it also showed the drop, like a decrease from the T0 to the flowering season and also the harvesting season. Again, we try to tease this apart, but I mean, it looks like it's obviously is very challenging because, as I said, the bean growth is come along with the season, also the application of the herbicide. So we have three things coming on in here, but you know, we still need to probably do further research, try to tease this apart and try to understand what the impact of each individual factor. So this is based on the, remember I mentioned like how many, more than 1.1 or 1.2 million sequences per sample, right? There's a lot of data in there. And to try to look at the community structure, we conducted this called NMDS plot, which is a non-parametric multidimensional scaling. And each dot here represents one sample. And keep in mind, you have 1 million data points in there. That's the species number and also the relative abundance, right? So how you read this, like if you have one data point right here and one here, they're pretty far apart. That means they are not similar. If you have two dots right here, and you have these two are close to each other, that means they're very similar. Here, I want to draw your attention is I have this circle here. So all these conventional samples, they are kind of clustered together. And all the organic here, which showed a circle, all the samples, it doesn't matter, it's green bean or black bean or which growing season, they cluster together, okay? On the left side, show the bacteria and the archaea. And on the right side, it's more clear. You see all the fungal community, all the fungi, the conventional on the left and organic on the right. So clear message there means, the community structure wise, the organic versus the conventional, they are distinct. So not necessarily to be like, you only contain the unique species, vice versa on each category, but the relative abundance or the community structure wise, they are different. So that's the first message we got. And we, of course, as I said, we are waiting for the year 2023 data. And when those data come in, we'll combine this data together and try to do further analysis. And another thing we also noticed that we have 4 replicates for this experiment. And also when we plot them, and here I separate the conventional organic, since from the previous analysis, we know they're different, right? So when we plot them separately, you see the conventional. Sure, you have the seasonal variations in there, but the samples from each field, they also cluster together. And what does that mean? That means, the spatial variation is huge, which is not a surprise, right? I mean, the soil, even like a 5 or 10 centimeters away, 10 centimeters away, they might be very different, right? So that's another challenging point for us is like, we try to collect more replicates. Hopefully we can cover the variations of the spatial variations. But, no matter what, the organic, conventional, they are different. But also when we look more detailed, when we zoom in, you also show the very strong spatial variations in there. So a quick summary for my part, the soil microbial community structures, and in this case, of course, we're looking at bacteria, archaea, and fungi, right? And in organic fields, they are very different from the conventional fields. And if you recall what Gladys showed you the results, and we suspect, and very likely, I think the microbial community structure are related to the bean quality, right? As Gladys showed, vitamins, the minerals, and the proteins, and the ergo. And how those microbes contribute to that? I mean, that's a different question, but we're heading to there. I mean, this is the first step, and we need to do further analysis, like try to understand why you have different microbial community structure And Diana is going to show some activities later on for the enzyme assay and how this related to the produced quality. So that's another question we probably need to tackle next. And also from our observation, we found that in general, the organic fields contain higher bacterial archaea diversity than the conventional, but the conventional, they contain a little bit higher fungal diversity. And also all these microbes, including both bacteria, archaea, and fungi, they vary in the growing stage and the seasons. So we know that because when you have a different season, different temperature, and in this case, a different growth stage, and also the application of the herbicide. Different changes in the temporal patterns in diversity and the spatial richness between these two groups of microbes, the bacteria, archaea versus the fungi, we notice that they are different. So potentially that means they may play different roles. And we know, in general, we know that they contribute differently to the nutrient cycle and nutrient uptake, right, for the plants. So we think they play different roles in the soil biogeochemistry and also which can be translated to the bean production and the bean nutrition. And then lastly, as I mentioned, looks like I'm glad we have 4 replicates for each treatment, but down the road, we may consider including more, or at least 4 replicates to try to cover our base, try to understand the big spatial variations. I think that's all I have, and I will give to Deanna, and she's going to introduce on the top of the microbial chemistry structure, and she's going to look at the enzyme assay, which is a function aspects of the microbial chemistry structure.
All right. I'll just then continue with our results based on the extracellular enzyme activity. So some of these results are for year 2022 and 2023. That will be what I will be focusing on. And I will start with a little bit of an introduction of why are we measuring extracellular enzyme activity and what it means, right? So perhaps you've heard of this type of metric before, but just to give you a review or a reminder, the extracellular enzymes are produced by soil microbes. Really, they're produced by all microbes, but in this case, in the soil, the microorganisms release the enzyme these enzymes into the environment in order to accelerate the breakdown of organic matter, the decomposition of the organic matter. And that facilitates then the uptake of carbon and nutrients that are part of the organic matter by soil microorganisms, right? So the microorganisms often release these enzymes as a response to nutrient demand. There could be other factors, but it's generally considered that enzymatic activity is a good indicator of soil metabolic activity, right? There's, of course, hundreds of different enzymes, and however, we have developed methods to be able to quantify the potential activity of some more commonly studied nutrients like nitrogen, phosphorus, and sulfur, and of course, carbon. So in our case, what we measured was beta-D-glucosidase, which it is associated with the acquisition of carbon, and acetyl-glucosaminidase, which is associated with the acquisition of nitrogen, phosphatase for phosphorus, and aryl sulfatase for sulfur. So how do we do this? We collected our soil sample from the VST, took it back to the lab, and within 24 hours, keeping it cold, we made a slurry at a pH that was similar to a soil pH. And then that slurry was incubated in a mix that contained the different substrates that, these are commercially available substrates that mimic the soil organic matter forms that the enzymes may be breaking down. So the breakdown of those substrates is equivalent to the process that may be happening in the soil, and then the production of the breakdown of the product of the breakdown of the substrate is fluorescent, and then you can measure that fluorescence using a microplate reader. So I should say, though, these are what we consider potential activities, right? Of course, they are not going to be exactly the same as what's happening in the soil. They are sort of like optimized conditions for the activity. So you just have to take that, into account. To get into some of the results, I will show you data for the 4 enzymes associated with carbon, nitrogen, phosphorus, and sulfur acquisition for 2022 and 2023. We have data for the green bean plots and the black bean plots. And again, there's 3 sort of periods: pre-planting, during flowering, or what we also have been calling mid-season, and then during harvesting. So the first thing I will mention is we didn't find significant differences between the green bean and the black bean soil samples for either of the enzymes. I'll maybe go back a little bit and describe the plots here first. Let's see, where's my pointer? Okay. So these are box plots. What you guys are looking at is basically the variation in the activity of 4 replicates that we measured in 4 field replicates that we measured in the lab. So the y-axis is the activity in units of micromole of substrate per gram of dry soil per hour. We have the data divided, comparing conventional inorganic, and then the different bean type. Black bean is gray and green bean is green. And then the 4 different enzymes. And so in either 2022 or 2023, we measure no significant differences among the 2 different beans, right? So I will only show you the results now for the green beans, because really they look very similar for the black beans. Okay, this is what I just told you. Now, looking at the green beans, we can look at 2022 first. Again, the plot is showing activity on the X on the y-axis. We have the 3 different periods: pre-flowering and harvesting. And there were some significant differences. The main observation that we saw was that there is definitely seasonality. Looking like during the flowering season is where we see the lower activities. And then there is certainly an effect of the system. So conventional versus organic. We generally see conventional soils having higher activities than the organic soils. However, that applies only for carbon, nitrogen, and phosphorus. It's interesting because sulfur is usually backwards. So in the sulfatase activity is actually higher in the organic fields compared to the conventional fields. So like I said, I'm not showing you the results of the black beans, but they look very similar. And actually, we saw also similar trends in 2023, where we see seasonality and activities being generally higher or lower during the flowering season, and then an effect of the system. So conventional versus organic, in many cases, we saw significant differences. All right, this is what I just told you. So to summarize our results, just keep in mind the measurements are really a potential metric, right? And if we interpret it as a descriptor of microbial and nutrient demand, microbial carbon and nutrient demand, we could say that carbon and nutrient demand seem to be not affected by not particularly affected by bean type, however, affected by the system type, so being organic versus conventional. Perhaps this is in part related to the content of organic matter, but perhaps it's also related to the different microbial communities, right? As we just saw, Jinjun showed us that the microbial communities are very different between the organic and conventional. And then perhaps the availability of nutrients could be also very different in the soil. And then that the activities of the enzymes are also affected by the growing state. So it's not clear. So there is a fact, the issue of the application of the insecticide, or the pesticide, sorry, but there is also, of course, the issue of temperature being different, and the seasonality might be part of the changes that we see from period to period. And then that the trends that we saw were consistent between years, which is always nice to see. All right, so that would be my part, and I'll get back to Gladys.
Thank you, Diana, and thank you, Junjun. I hope that you had a great webinar with us today, and I want to acknowledge our collaborators and funders. We have been working with Dr. Zachary Pitzer from Penn State University, who is helping us with the ergothioneine and developing protocols for different crops. Also, Dr. Lavanya Radivari from Purdue University. She is also working on the anthocyanins. So I wanted to thank our collaborators and the funders, Grantham Foundation, Department of Agriculture of Pennsylvania, also the Orange County Community Foundation. And not to forget the big thank you for our research team, the field operation team, and the research interns that work with us. And at the end, thank you. And if you can, and if you have any questions, you can ask now. You can put your questions in the question answer. And here are our emails in case you don't have yet any question on your mind, but you want to follow up with us. I want you to put any information you need in the question and answer. There was one, it asked about if we did add any rhizobia. And yes, every time we plant green beans or black beans, there should be inoculation with rhizobia for any beans we put. Even if your soil, you think it has that rhizobia, it is always better, especially because we are doing research. We inoculate prior to the, prior to the planting. Let's see, there was another question, if I can find it. For the conventional fields, which ended up with higher fungal diversity, which fungal families were present? Junjun, do you have any answer for that?
Yeah, I cannot remember at the top of my head, like, there's a family members of quite a members of family in there, but I do have some simple plots in here. I mean, I cannot summarize to the family level, but this is on the phylum level. We have the most abundant ones, Ascomycota. Yeah, so yeah, that's a good question, and I just cannot. I mean, that's you can imagine the data we have contains not as many as the species name, but the families are quite a bit, right? I mean, here we have the major phyla including here for both organic and conventional. This is the not exactly the same, but it will have similar kind of observations, and that's what I'm talking about like, we have this family members or the species names, and what next step to do is like try to identify the indicate species for the organic and the conventional. If you want to do that, I mean, like not only the phylum level, but all this species level depends on what level you want to go to, right? So this different family, I mean, I cannot remember on top of my head like which one is more abundant or more than the organic or vice versa. But these are the three major phyla we observed for the fungi.
Joe Tobias says, Ascomycota in convention. Do you have any speculation as to why this group dominated the system?
I don't have a good reason. I mean, what we observed, I mean, you can tell from both sides, organic and the conventional, the Ascomycota. I mean, my understanding is always like a predominant fungal phylum in the soil, and obviously they are doing a lot of stuff for the plants. And I don't know. I guess with the current knowledge, we don't know quite why these groups of fungi they are in there. I would assume they are helping, with all this organic matter. I mean, the Ascomycota, they are really good at organic matter degradation, for example. I mean, when you have all those organic matter in there, they are helping with, degrade all those, and then with working with the bacteria together. So that's kind of like I guess another part is like we probably need to look at only this one group. We need to look at them together. So sometimes they have this beneficial relationships with all the other microbes. So I don't have the specific answer for you, but I guess my guess is the abundant in both systems, and they might be helping the plants and also help with the other microbes working together for the soil health.
Thank you. I don't see any other questions in the. There is another one in the chat. Says, does the increased nutrient quality in organic produce reflect findings from other studies? How do you anticipate this will influence public health dietary recommendations? That's from Sapna P. We have seen in other crops in the vegetable systems trial that nutrients between different crops vary, meaning that while we see sometimes, let's say, proteins, vitamin B6, phosphorus, or magnesium are greater in organic than the conventional, let's say, in the squash, but we see also that the other crops may be showing no significant differences, but like potato, but we can see that all their nutrient densities are really high, and maybe potato is not as high in calcium like winter squash, but the trend of having nutrients, especially proteins, in the organic system, and even the amino acids are greater in the organic crops than in the conventional. How that it will influence the public health, that's a choice that they can make, the public, that they depend on what is good for them. We can only suggest and recommend that for resilience, for immunity, to improve your resilience against any diseases or future issues, such as when people aging. It is always helpful to choose the crops, the produce that it is rich in nutrient density and grown properly in a way that has less or minimal pesticides or herbicides. And that really, because every of those chemicals, they could affect your kidneys or your heart or your digestive system, which is the most important, where the microbiome is, that really releases all these enzymes to whole body. So it is a choice that the person will make, the public has to make based on what is good for them and what they want to see as a result from that food. So how that will affect dietary recommendations, meaning, in fact, this project that we started 6 years ago, the vegetable systems trial, is made specifically to answer so many of those questions without a bias. How, when you produce a crop, side by side, the organic and conventional, are they going with time showing a effect on the nutrients? Is the nutrient gonna be different with time? This also, the practices that we are using, organic versus conventional and tillage and reduced tillage, how they are managing the soil health and how they are changing the soil health. Last week, I presented data for you. If you go to the webinar, it's already uploaded. You could see that in 6 years, we have increased the proteins in the soil. We increased the labile carbon, organic carbon in the soil, in the organic system. We increased the carbon and the organic matter within 6 years. And the organic matter was increased by 1%, which is incredible. But we also saw that we have a decline in the conventional. So any soil that is really treated not organically, and even if it is organic with tillage, with time, these microbes will release their nutrients. They will do a different cycling, and the plants may not be as resilient to climate as those that they are under reduced tillage and organic. Even the conventional with reduced tillage, you could also minimize the carbon. You can minimize the organic labile available. You can reduce the protein with time. And that down the road, you will see the changes in the nutrient density. Was compost utilized in the organic system as an input? When we grow beans, we do not apply any fertilizer in the organic or in the conventional, because usually it is a carryover from the crop that it is there. So we only put the cover crop and we just roll it or spray the herbicide, and then we grow the beans. Now, the previous crops, they would have amendments in them, either organic or it is a conventional, which is synthetic fertilizer. But during the season, we do not do that. And that's all. And I think we are done. Thank you. Bye-bye.